Hi there. Could you tell me a little bit about yourself?
Yeah, hello. I'm Mohammad Alfatah, and I'm a senior research fellow at HLTRP, Healthy Longevity Translational Research Program, NUS Medicine, Singapore.
And where are you mostly based?
I'm originally from India, and I'm currently based in Singapore, and I'm running my own research group under mentorship of Professor Brian Kennedy.
What does a typical workday look like for you?
Supervising my students, doing some experiment, like here I'm going to do some experiment and testing some compound for zero protection, and writing the manuscript and the grants, and sometimes quite a rush between the experiment and the meeting.
So what's your research about?
Yeah, I'm trying to identify the chemical genetic intervention that delay the aging and increase the lifespan. So I'm using the very powerful model system Yeast for cellular aging experiment. And here, for example, I just show that we have cell wild type and the mitochondrial mutant and different kind of the mutant. And I've already aged with different chemicals and natural products and FDA approved drug. And I'm going to test how this compound can protect the problem of the mitochondrial dysfunction, or maybe can increase the lifespan of the wild type cells. So this all high throughput system we have established recently. And today is day 7, and I will show you that this is already aged plate. So day 7, and typically yeast survive for 10 to 14 days. So I'm gonna check the survival, and how we check the survival using the high throughput system. And I just show you here. So here we can see that we're going to take some cells, which aged cells, and here I will spot it to my culture. We took here and we'll transfer to the fresh media. And this one too, we will incubate at their temperature 30 degrees Celsius, and tomorrow we'll see whether cells are surviving or not. So any compound, for example, rapamycin, we have established that rapamycin always protect the mitochondrial dysfunction and increase the lifespan. So again, I will incubate this plate at 30 degrees Celsius. And tomorrow, the growth and the survival will be read by spectrophotometer.
That's so interesting. Yeast, I never knew you could use yeast in science. So what makes yeast such a powerful tool in understanding aging?
It's not quite new, because the yeast basically Saccharomyces cerevisiae is contributed huge in biomedical sciences. For example, most of the discovery of aging hallmarks like mTOR, nutrient sensing, and mitochondrial dysfunction, and autophagy, and telomere, this all actually discovered in the yeast. So that's why yeast is a powerful model system, and that is a very simple eukaryotic model system. And why we use for chemical and high throughput screening, because they have a very short lifespan, like 10 to 14 days, divide very fast, quite genetic tractable. So that's why, and most of the biological process and the aging hallmarks quite conserved between the yeast to the human. That's the reason that we are using the yeast as a model to identify the chemical as well as genetic intervention that delay the aging and increase the health span and lifespan.
And when you're not busy studying yeast, what do you enjoy doing outside of lab work?
Yeah, I spend my time mostly with my family and my friend. Yeah, and recently I have some kind of new hobbies that I'm interacting with some aged people actually in my community, and I'm trying to understand after I'm joining the HLTRP, because here, for example, I know that people are actually suffering like cognitive problem, cardiovascular diseases, and cancer. These all associated with the aging, and I'm trying to understand how they feel during when they are aging, what actually they want, and what they want to enjoy, and what they want to eat, whether they are eating the cake things or not. And we are the scientists, we can help to let them know that, okay, these things could be good, this thing could be bad, and like, for example, supplement nutrients, because we can see very in depth, right, that how this FDA approved drug and supplement is beneficial, not beneficial. So I think they require our attention, and we should talk to them. Yeah, if you get the time. Yeah.
Absolutely.
Yeah.
And I hope you get to spend more time with these precious souls. Maybe you guys can bond through Yeast, like making bread together.
Yeah, sure. I'm trying it.
How can we follow you for more?
Yeah, you can connect me at my email. You can see here, and if you want to know more about what we are doing on the aging biology, if you want to screen your some compounds, chemical that you find that could be potential genoprotective compound. For example, recently we discovered curcumin, erythromycin we are using as a standard gold, and we're also trying to find some intervention like that combined together and with rapamycin, and they further synergize the problem. So you can just connect me at my email ID.
Amazing. We'll see you later at the motivation.
Yeah,
See ya.
thank you very much for listening to me.